All right, so this is where it's going to get a little bit harder, I think. We're going to look at chemiosmosis, but before we do that, we need to look at the electron transport chain. So there's really three terms that are going to be a little confusing on which ones to use. Okay, so what I need you to know, the electron transport chain is basically a structure. It's a bunch of proteins and other molecules that are set up, and they are embedded in the mitochondrial inner membrane. All right, so here's what I need from you. I need you to know there's really two here, and they're kind of hard to see. So what I need you to know is one chain is FMN, which stands for flavomononucleotide. That's a protein, which is passed to a molecule called iron sulfide, which then gets passed to Q, which is ubiquinone. Why it starts with a U and they're using Q, I don't know. Okay, ubiquinone. I don't think I said that right. Okay, so ubiquinone. All right, and then we have what we call the cytochrome complex, and that's all of these molecules here. Okay, so let me say that again. So there is one chain that's FMN, flavomononucleotide, goes to another molecule, iron sulfide, which goes to ubiquinone, and then a cytochrome complex. The other one goes directly to iron sulfide on a different molecule, a different protein, but not that important. And then it gets passed directly to ubiquinone, and then goes through the cytochromes. This is going to become important later, and so I'm going to explain it now. What happens is we've made coupons. We've made NADHs and FADH2s. So what we want to do is look at those and talk about what that means for us. Okay, so when they make the NADHs, what happens is they can be attached to the FMN, they run down this chain, and that's going to pump enough hydrogens during chemiosmosis to make about 3 ATP for each NADH. FADH2 is actually put on the kind of on the slide a little bit lower. It goes to the iron sulfide. It's actually a different iron sulfide molecule, not important. But because it's put a little bit lower, what you're actually seeing is it only makes 2 ATP for each of those. So it's pumping enough hydrogen ions to actually make 2 ATP. So again, each NADH can equal up to 3 ATP, and every FADH2 can equal up to 2 ATP. You kind of remember that with the 2 being 2 and NADH being a little bit more. So again, NADH hooks to FMN, flavomononucleotide, FADH2 hooks to iron sulfide. All right. All right, and then here's the ATP synthase. We talked about this kind of before. We didn't, and I don't care about all the little parts, but this is what's part of that chemiosmosis. So I want to show you that again. We saw this with photosynthesis happen on the thylakoid membrane. This one's now happening on the inner membrane of the mitochondria. And so what's going to happen here is we're moving things across, and what's happening here is NADH gets attached, and we're pumping in hydrogen ions into the intermembrane space. And NADH again is pumping enough hydrogen ions to make 3 ATP. FADH2 is pumping enough to make 2 ATP. And these hydrogen ions are sitting over here, and then they want to come back. And as they come back, so we're going from the matrix into the inner membrane space, and then coming back into the matrix. And so what you're seeing here as we're doing this is we're producing ATP. That's the whole purpose. Here's our ATP synthase. The hydrogens can't go through the phospholipid bilayer. They need a transport protein that happens to be ATP synthase. All right, so what else do I need to tell you? Thinking about this is that let's give you the terms. So the electron transport chain is basically the structure. Chemiosmosis is really a process. It's pumping the hydrogen ions across and letting them flow back through. And then technically oxidative phosphorylation is adding this phosphate. And what I haven't told you yet is this is driven by an oxygen molecules. Oxygen molecules are very negative. They love electrons. And so the reason that the electrons travel down this electron transport chain is because they are attracted to oxygen. The reason they are driven in chemiosmosis on the other one is because of light. Okay, so that again this is called oxidative phosphorylation. The other one in plants is called photophosphorylation. Okay, so kind of a complex, this picture is very similar to the picture we saw in photosynthesis, but a little bit different. Okay. All right, so overview, and we're going to talk about this in lecture when we're together. Maybe this week, maybe the following week. We'll see how that goes. All right, so we'll talk about maximums and stuff later. The last thing on this, we'll talk about several different things that are going on. Now, one of the things that we haven't told you is the reason you need to breathe. It's because you're stressed. No, the reason you need to breathe. The reason we need to breathe is to take in oxygen. The reason we need oxygen is cellular respiration. Everybody goes, oh, we need to breathe to live. Well, it's to make ATP. And that's called aerobic. But there are some organisms anaerobic that can actually make ATP without oxygen. And that's what the process of fermentation is. It's an anaerobic process. And so what you're going to see is alcohol fermentation is actually without oxygen. And so what happens here is pyruvate does not necessarily go into the rest of cellular respiration. So what happens though is it needs ATP. So it's a two-step process. I don't really want you to worry about it, but what you end up with is two ethanols, which is the alcohol, and two CO2s. And that's how you make alcohol. Alcohol has to be made anaerobically. You seal it. And so we can't do that. Probably a good thing. Holding your breath is not a way that you want to get drunk. And so this is seen in yeast, seen in bacteria, alcohol fermentation. All right, the next one is something that does happen in us, and this is called lactic acid fermentation. And what you're seeing here is we're producing pyruvate. Again, doesn't go into the Krebs cycle and transition phase. It is converted to lactate or lactic acid. And what happens there is that will build up. So we're getting our ATP. You can see that's still being produced. But then lactate is typically in and this happens in our muscles. And at one point, they thought the lactic acid buildup in your muscles was actually what was causing pain after you worked out. Turns out we don't believe that anymore. Actually, that lactic acid actually tends to help the process in your muscles to work better, and that lactic acid gets funneled off to other cells to later be broken back into put back into glucose, glycogen, or broken down further. And so what happens for us is we believe the pain in your muscles is not from lactic acid buildup, but actually from the micro tears that happen in the fibers, which cause pain and cause inflammation, which eventually goes away in a couple of days when all that heals itself. All right, reality. Here's what I don't know you well. Maybe you do eat pure glucose. I'm doubting it. But I want to make sure you realize that that's not necessarily the case. I want you to realize everything you eat actually ends up in, or can end up in this cycle. And I'm not even going to ask you where it fits in, but I want you to know carbohydrates or sugars can pop in the beginning of glycolysis, or actually jump into some of the first 5 steps. The investment phase of glycolysis. And then I want you to know that proteins will actually can be broken down into pyruvate, can go in as acetyl coenzyme A, or actually go in as one of the other parts in the Krebs cycle or citric acid cycle. And then fats can go in as G3P and or acetyl coenzyme A. So what I really want you to know is everything that you eat, all those things can go into this process to make ATP somewhere. And then I want you to know what controls this system in your body. And so there's these feedback mechanisms, and there's 2 things that actually inhibit the making of ATP. One is excessive citrate. If you have lots of citrate, that'll kick back up and it'll shut the sucker down in the glycolysis process. Or if you have a lot of ATP, you don't need any more, same thing, it'll shut it down. So ATP and citrate both are triggers to shut down the process. What I want you to know is AMP actually stimulates the process. And what AMP is, okay, so we have ATP, which is adenosine triphosphate. We have ADP, which is adenosine diphosphate. But we also have AMP, which is adenosine monophosphate. So again, we have those 3 phosphates on the end. We spend most of our time breaking that 3rd one off and putting it back on. But if your body starts to struggle and you need energy, actually ADP starts to get broken down into AMP. And if you have a lot of AMP, obviously your body is starving for ATP. And so that will kick this cycle on. And with that, we will stop the share and stop the recording.